I'm Dr. Gloria Jimmy Cristobal, Assistant Professor, Department of Biotechnology, V.V. Vaniya Perumal College for Women, Viruthinagar. So this video is going to educate you about cell culture, cell lines and organ culture. So cell and organ culture techniques are essential tools in biotechnology and biomedical sciences, providing a controlled environment to study the structure, function and behavior of cells and tissues outside the living organism. These methods help researchers stimulate physiological conditions, enabling advancements in drug development, cancer research, genetic engineering, vaccine production, and tissue regeneration. Understanding the types of cell culture, that is primary, secondary, and organ culture, is fundamental to explore cellular mechanisms, disease models, and therapeutic strategies with precision and reproducibility. So coming to cell culture, we have two types of cell culture. One is adherent cell culture, another one is suspension culture. So in adherent cell culture, cells grow attached to a solid surface, for example in a culture flask or dish or in a petri plate. So it requires surface for attachment. For example, fibroblasts and epithelial cells are cultured as adherent cultures. So they may be cultured either as primary culture or secondary culture. Next is suspension culture. So here the cells grow freely in the culture medium. It doesn't require any attachment surface. Examples, lymphocytes and certain tumor cells and hybridoma cells are cultured by these means of suspension culture. So here also primary culture is also possible and continuous culture is also possible. So so here you can see this is the suspension culture and it freely floats on the medium. And this is the adherent culture, so it is attached to the surface. Mostly the culture flask will be coated in fibronectin. So the adherent culture, it gets attached to the surface of the flask. So coming to the primary cell culture, it refers to the process of isolating cells directly from the tissues of a plant or animal and culturing them in an artificial environment for the first time. So what are the characteristics? Is the cells, they closely resemble the parent tissue in morphology and function. They show normal diploid chromosome number and it has limited lifespan. They undergo senescence after very few divisions. Heterogeneous population, they are heterogeneous in nature, and that is they contain different cell types. So they are high sensitivity to contamination and require strict sterile conditions. So this is one of the primary cell culture method. So from the rat, the gland tissue is removed and it is minced, and after adding collagenase, it is ground and it is made into pellet. So then it is the pellet is washed, and after centrifugation we can introduce different cell types into the culture flask. So what are the techniques of primary culture are? First one is mechanical disaggregation. So in this method, it is a physical method where the tissues are broken down into small fragments using tools like scalpels, scissors or forceps. And the procedure is first the fresh tissues, it should be washed with sterile buffer to remove the blood and debris. And then it is cut or minced into small pieces using sterile scissors or forceps. The pieces may be further suspended repeatedly, pipetting up and down to separate the cells. Then the suspension is filtered through a cell strainer or mesh to remove large clumps. The resulting cell line suspension is centrifuged and resuspended in the culture medium. So what are the advantages are? It is simple and quick, no need for any enzymes or chemicals, it is low of cost. Next method is enzymatic digestion, where we use different enzymes to digest the extracellular matrix and cell to cell junctions so that the individual cells are released. So what does the procedure means? The tissue is minced into small pieces, incubated in the enzyme solution. We will see about what are all the enzymes used in the upcoming slide. First, we can use either trypsin or collagenase at 37 degrees Celsius, and then the digestion is stopped using some specific inhibitors, and then it is filtered and centrifuged to collect the cells. Then the cells are resuspended in the fresh culture medium. So what are the advantages? They yield high number of viable cells. The previous method we may face some cell death, but here the number of cells, viable cells will be high. Then it is suitable for fibrous or dense tissues, and then less physical damage compared to mechanical methods. So these are the common enzymes used for enzymatic digestion: trypsin, collagenase, displase, hyaluronidase, and then DNase. Next is explant culture. So here a small tissue fragments, they are taken in the culture dish without prior digestion or disaggregation. The cells then migrate out of the tissue over time. So it will take some time, but this procedure is very effective. So coming to the procedure, a tissue is cut into small pieces under the sterile condition, and then the explants are placed in culture dishes, allowed to attach for a few hours in small volume of medium. After adherence, more medium is added to support the cell growth. Over days, it may take some 1 week or 10 days, it will migrate out from the explant and begin proliferating. Once a sufficient number of cells grow out, they can be trypsinized and it can be subcultured. So the advantages are simple, they are very simple and enzyme free. It minimizes cell surface damage, and then it is useful for slow growing cells such as nerve cells. So coming to the examples of primary cell culture, we can culture primary hepatocytes, primary neuronal cells, primary keratinocytes. So the applications of primary cell culture is we can use for drug toxicity and metabolic screening, studying cell signaling and gene expression, virology and vaccine testing, cancer and immunology research, tissue engineering. And then advantages, they are closest to in vivo condition, that is it resembles our body condition, and then it yields accurate results. So disadvantages are the availability is very limited and it grows very slow and it requires ethical permissions, and it is prone to contamination and genetic drift. So next is secondary cell culture. Secondary cell culture is derived from primary culture by transferring cells to a new growth medium to expand the population or maintain viability. So the key characteristics involve homogeneous population compared to primary culture. It may undergo slight morphological and functional changes over time. It can be passaged only for a limited number of times. So it has very finite lifespan. Usually more stable and easier to handle than primary cultures. So passaging process, it involves cells, it should reach confluency. For removing, we should use trypsin or EDTA so that it can get detached, and then we have to transfer it to the fresh new flask and fresh medium. So the examples, it includes human lung fibroblast, which is derived from primary tissue using viral vaccine, and then mouse embryonic fibroblast. So applications, it involves scaling up of primary cells for experimentation, genetic manipulation and transformation studies. So cytotoxicity and carcinogenicity testing can also be performed using secondary culture. Protein expression and assay development can be performed using secondary culture. So the advantages, it includes it is more uniform and it is reproducible. It is available for many number of cells are available and it can be used for large scale work. So disadvantages, it includes gradual loss of original characteristics, finite number of divisions and risk of contamination or phenotypic drift. Next, coming to cell line. Cell line is a population of cells that has been subcultured multiple times and can grow indefinitely under in vitro conditions, especially if it is continuous cell line. So types of cell lines, it includes finite cell lines and continuous cell lines. So finite cell lines, it is derived from normal tissues. It divides up to 20 to 80 population. It doubles before senescence. And continuous cell lines, it includes transformed or derived from tumor cells. So the key characteristics of cell lines are it is homogeneous in population and it can be genetically modified. Often it is used for long-term experiments. Easier to maintain and handle, that it can be handled very easy than the primary cells. So what are the examples of finite cell lines means? WI38, which is human lung fibroblast, MRC5, human embryonic lung fibroblast. So the examples of continuous cell lines, HeLa cells, which is human cervical cancer cells, and CHO, Chinese hamster ovary cells. This is used for the production of recombinant proteins. And Vero cells, which is from African green monkey kidney, it is used for vaccine production. And 3T3 cells, mouse fibroblast line, which is used for cancer research. So the applications, it includes cancer and genetic research, and then biopharmaceutical production like monoclonal antibodies, vaccine production, and it is used for studying drug screening and cytotoxicity testing, and it is used for virology and recombinant protein expression, stem cell biology and gene therapy. So the advantages of maintaining cell lines, it includes unlimited supply of cells, stable phenotype, and easy to standardize. And it is well characterized and it is reproducible. So coming to the disadvantages, it may differ significantly from the tissue of origin. Always we know the in vitro condition is completely different from the in vivo condition. And a genetic drift, that is some genetic change, will surely occur over a very long period of time. And then the risk of contamination is very, very high, especially due to mycoplasma. Okay, students. So thank you for bearing your time with me.